Welcome to Therapeutic Momentum. We take you on an inspiring journey, spanning breakthroughs in science and medicine development. Over the last century, our average life expectancy has increased by more than 20 years. Join us as we go behind the scenes with medical researchers, clinical developers, and other experts working together in the race to tackle life-altering diseases with new treatments and cures.
I'm Karen Akinsanya. I'm a translational scientist working at the intersection between biomedical research and emerging technology. Thanks for tuning in to Therapeutic Momentum. Let's get started. It's my great pleasure to welcome Andy Plump. Andy is the president of R&D at Takeda, where he leads the company's global efforts in drug discovery, translational medicine, and clinical development. With a career spanning both industry and academia, Andy is well recognized for his strategic vision in integrating cutting-edge science with data-driven decision-making to accelerate the development of transformative therapies. Andy has overseen strategic and cultural transformation since joining in 2015. He's also played a key role in reshaping the company's R&D strategy, fostering a culture of external collaboration and innovation. Prior to Takeda, Andy also held senior positions at Merck and Sanofi. Andy, just reflecting, I think it's 2005 when we first met. I think of you as one of the most energetic people that I've ever worked with such a sense of urgency. I learned a lot from you at Merck, and I'm really happy to be sitting with you to hear more about your journey, how you got into pharmaceutical research. You're a trained physician, cardiologist. Maybe you can take us back to how that happened. Tell us more about yourself and what motivated you to develop drugs for a living.
Well, thank you, Karen. It's fantastic to be here with you. I can't believe it's 20 years ago when we were sitting in that conference room in Merck, and you were in clinical pharmacology, and I was in the cardiovascular discovery group, and I was trying to recruit you over. Remember that conference? Do you remember that conference?
Yes, I do.
It was great. We've had a long history together, and it's really great to be back together again. So I joined Merck in 2001. Actually, my start date was October 11th, 2001. Oh. And I was actually soul searching that year because I ended up doing something in my postdoc. It was very fundamental biology. I was studying axon guidance in the developing central nervous system with Mark Tessier-Levine at UCSF, and I loved it. But I had felt like I had gone down a bit of a rabbit hole because my interests were not per se in basic fundamental biology. They were really in translational medicine. In my mind, I had reasoned that disease are just developmental biology pathways gone awry. So if I could study developmental biology, I could then rapidly translate that to disease biology. And that gap, though, is massive. Mm-hmm. So as I started to work more and more on studying neurodevelopment, I started to realize I was moving more and more away from what I really loved. And so I was going through soul searching, and then as is almost always the case, it was somebody that helped to pull me back. And it was Tino Melian. Oh. Do you remember Tino?
Yes.
He was in the I&I group with Elise Ricin and Briggs Morrison, really just an amazing cohort of people. I ran into Tino. We had lost touch. We had worked together in a lab many, many years prior. Very similar mindset, similar background, and he was at Merck. So he invited me to spend a few days there, meet with, I must have met with 30 or 35 people, and I got very excited.
Merck has a way of interviewing, having them meet just a huge number of people. I remember when I went for an interview, actually I went to an interview in Pennsylvania and in New Jersey.
When was that?
2004, 2005.
Okay.
And the list of people I met, it was just a full-on day, and it was amazing, wasn't it? Just really exciting.
It was amazing. I remember at the time Merck had this system where employees who worked their lifetimes in the factories could actually apply for a chauffeur position. They had 4 chauffeurs. These are Merck-owned cars, Merck-owned chauffeurs. Yeah. And there was one very famous chauffeur who was actually written about books that have been written on Merck. He's quoted in these books. I don't remember his last name. His first name was Guy.
Oh, right.
And I get into the airport at Newark for this 3-day interview, exactly to your point, meeting with 30 different people. And I just spent the 30 minutes in the ride listing out everybody that I'm meeting with, and he knew all of them.
Ah, yes.
He was giving me the skinny on all of these people. It was very funny. So
That's amazing.
But I visited, and then I reasoned that I needed to expand my horizons. I shouldn't just be interested in Merck. But the reality was I was quite impressed by the people that I had met at Merck and the history of Merck. And it was, for many, many years under Roy Vagelos, it was not just the best biopharmaceutical company, it was the best company to work in across all sectors. And so I started looking and talking with a lot of people. And actually on September 12th, the day after 9/11, I was actually supposed to go to BMS for an interview. And of course that got canceled. And, I basically pulled back from a lot of the soul searching I was doing because I just loved what I had seen at Merck. And so that was my first foray into the biopharmaceutical industry.
So you joined the I&I group, or you joined in CVM, or?
Well, so I interviewed in 5 different groups. The I&I clinical group, clinical pharmacology, discovery research. There was a new group that Sean Harper had just started to run, which was pharmacogenomics. And then, oddly, the one I was most interested in was project management.
Oh.
And you remember at Merck, Hamish Wright, who we were just talking about, Bill
Yes.
Jew, these were physician scientists who
Right.
were in project management.
That's true.
And I thought that would be the perfect way to holistically bring my clinical in. So in the end, though, I decided clinical pharmacology, just the same as you, because it just felt like, when you train in medicine, people always wonder how you eventually decide what subspecialty you want to go into. And more often than not, you see yourself in the people who are doing that subspecialty. And that's what happened with me with clinical pharmacology. I saw a group of translational physician scientists who had backgrounds much like I did and who thought a lot like I did.
Yeah. And the pace in Clin Pharm is just incredible, isn't it? Just new molecules all the time, trying to figure out how do we make decisions about the viability. It's such a dynamic space.
It was amazing. I learned so much. At age 35 when I first started in Clin Pharm, and I thought I had learned something over the last 15 years of my training, and I came in and I realized I was almost starting afresh.
Right. Every project, right?
Yeah.
There's something new to conquer. So you started in Clin Pharm. I don't know whether you'd already moved on to cardiovascular. Yes, you said it. You were already then in cardiovascular,
That's right.
I think. And we ended up working on a protocol together. I don't know whether we're allowed to say the mechanism.
I think we can 20 years later. It was
I think we were trying to figure out how to measure some kind of evidence that there was change in plaque, right?
Yeah.
I vividly remember writing that protocol and getting your feedback. And so thinking about cardiovascular, pretty tough space, but you were leading that space. And I think one of the things I recall was your views on genetics and how important that was. Looking back, have we made the progress that we had wanted to? Has the genetics paid off in leading us to new mechanisms?
Yeah. So it's interesting because if I fill in a gap, actually. So I left Clin Pharm 2 years into my tenure at Merck to join a new group that was working on pharmacogenomics.
Oh, you did?
And we actually were working with Decode at the time. Amgen eventually bought them. And so I stepped into that group and learned a lot. We didn't really get a lot done. It was a hard time. We had a hard time penetrating difficult space and a hard time penetrating the development wall within Merck, which was quite outstanding, but also quite traditional. So then I ended up leaving, and I went into a translational role in cardiovascular, which was my sweet spot. I loved it. And if you remember this, but Luciano Rossetti was running kind of the broader group, and was having difficulty bringing in a new discovery head to replace Sam Wright, who had left. And so he asked me if I would be willing to step in, and it was one of the most difficult professional decisions I had to make, because I loved that translational space, and I didn't really have a good sense for what the discovery space would look like. We're scientists, but there's a difference between being a scientist and being a drug hunter.
Right.
And I definitely was not a drug hunter at the time. I was a scientist, and
Right.
So I took it, and it was by far the best move I ever made in my professional career, moving into that space. And so we worked on atherosclerosis, we worked on hypertension, which is a group that you came in to eventually lead.
Right.
And then we worked on thrombosis. And, it was before things dipped in cardiovascular. It just everybody left cardiovascular to go into specialty care and into rare diseases, and it's now come back. But your question, have we made the progress that I would have imagined? I think yes. And I think we've really realized so much of the opportunity that genetics has provided us in cardiovascular disease. I mean, it's just amazing if you think about it. I think the issues now in cardiovascular disease are around overall healthcare. It's a little bit less around novel therapeutics, especially in some of the primary indications.
Yeah.
And then heart failure.
Yes.
That's the big open space right now.
Yeah, you're right. I mean, factor 11, I remember some of the genetic stories there, and rare alleles of large effect versus your chronic large population. Was that going to translate? And I think we've seen now that is the case, right? Yeah. And we were hoping to look for more signals like that. I actually was talking to George Davey-Smith recently, and I remember we had all these fantastic ad boards with the geneticists, and obviously we were working with Sek Kathiresan. I think we ran the first exome array with Sek when he was still at the Broad. Yeah. I certainly believe that if you look back retrospectively now at some of the mechanisms that have made it in the clinic, they often have this strong underlying genetics, either as those rare alleles, but also potentially signals coming from more GWAS and so on.
For sure. Even the statin target, HMG-CoA reductase, which was not, I mean, of course it was a genetic mechanism because of what we had known from familial hypercholesterolemia, which is an LDL receptor deficiency, and right. there's a strong interplay between LDL receptor, intracellular cholesterol loading, and HMG-CoA reductase. But at the time, there was no kind of genetic correlate that linked HMG-CoA reductase to cardiovascular disease in any way. With the GWAS studies now, you see signals in HMG-CoA reductase, and they're real.
Right.
So, it was an amazing journey. The hard part, of course, was finding mechanisms through genetics that could differentiate beyond the outstanding drugs that existed. And as you mentioned, factor 11 is a great example of one where we're now seeing that.
Yeah. I would say, looking at your career, you obviously worked at a huge US-based company like Merck, but you've also worked for some globals. You went from Merck onto Sanofi. Maybe you can tell us more about that transition. And then more recently, obviously, gone from Sanofi to Takeda. Mm-hmm. That gives you an interesting global perspective on the pharmaceutical business, what it's like to work in global organizations. Maybe you can tell us more about that and how you perhaps have evolved as a leader with that in mind.
It's been an incredible journey. And having been through 3 companies, one of each, basically, in terms of culture. The learnings have and continue to be just amazing, and I've grown so much as a human being and as a leader, and I continue to grow. You can't stereotype a company per se, but Merck had this incredible discipline and scientific excellence, and there was the Merck way of doing things, and it was pretty good. It was both enabling and created a barrier. It was enabling because it was a recipe for success, particularly around small molecules. It challenged Merck's ability to move from small molecule chemistry to other modalities, because the mindset shift was a difficult one for Merck to make. Mm-hmm. Sanofi was an interesting transition. It was funny because when I left Merck, nobody at Merck could understand how I could leave Merck to go to Sanofi. And then they started to realize, Andy will move to Paris. And so Paris they could understand, not so much the company shift. But Sanofi Sanofi was a really young company. Merck was founded in the 19th century, and Sanofi was a mid-20th century company. So it was a young company that was incredibly successful, that was trying to still find its way. And as with many companies at the time, this was 2012, there was a transition away from small molecules to other modalities, and had just bought Genzyme. So I learned a lot at Sanofi. I learned a lot about being a global company. I learned a lot about being open to new ways of thinking, in some respects different from Merck, and then rare disease and specialty disease. Merck had been very much a primary care focused company. And then, of course, the third transition, which was I didn't spend enough time at Sanofi because this opportunity at Takeda came around in 2014. And so this last opportunity working in a Japanese company, when I joined in 2014, 2015, the board meetings were 100% in Japanese. There were 13 board meetings a year, all in Japan. 50% of our business, 50% of our employee base was in Japan. And so truly a Japanese company, but a Japanese company that was primed to globalize based on decisions that the prior CEO, Yasuo Asagawa, had made, and that Christoph Weber, our current CEO, was brought in to help to catalyze. So I came in, I think one of my intrinsic, I'm a middle child of 5, so intrinsically I like to bring people together, and then having that entrenched in the global company that I was in Sanofi helped me then step into this job at Takeda and bring those cultural sensitivities and understanding as we started to then expand Takeda into a global company.
Wonderful. So you've actually been quoted on culture. You believe that culture is not something you put a slogan on the walls, but it's much more than that in terms of how you not just create a great culture, but maintain it. Do you want to share some thoughts there?
Yeah, I will. And then I'm curious in your thoughts on culture, because I'm sure you've, in your experience at Merck and now at Schrödinger, you have similarly learned an immense amount.
Yeah.
So it's funny because when you talk about culture, of course, it invokes a different reaction depending on who you're talking to.
Yeah.
But in the corporate environment, there is a way of interacting. So companies do have their kind of cultural tendencies. Some cultures are very direct, some cultures are very deferential, some cultures are nice, and there are confounding factors that come with each of those, right? That either enhance or impede your performance. But culture is not just how you treat one another, it's your way of working.
Yes.
Right? How do you work? Do you work through consensus? Do you work through team? Do you work through top down, right? So these are really important, and it's so important that you understand the context that you're within, because you can change process, you can change your portfolio, you can change your strategy. Culture is really hard to change.
It is.
And the only way that you can get in there and move, you can't change it overnight, but you can move it, is by deeply understanding the cultural context that you're within. I mean, have you seen this?
I have seen it, and I will reflect on something that you said. Now, Merck is this incredibly sort of organized, high-quality organization, but I think there are also some subcultures, and one of those subcultures that I fell into was working with people like you. Very dynamic, trying to push the boundaries. You talked about moving into new spaces. And one of the things that has stuck with me is the ability within a sort of Uber culture to foster these subcultures where people are trying new things and they're not afraid and they're pushing hard and they're bringing in early career people. When I think I first met you, I had come from Ferring Pharmaceuticals, a very old, very traditional company as well. And there was this dynamism that you brought, I think, to really keep pushing those boundaries. Bringing an early career person in and giving them some freedom to operate, I thought was immensely important. And I've carried that with me. And so now a cultural cornerstone for me is my leadership team has very early career people on it who are challenging me and pushing me and not letting me sort of settle into we've always done it this way. And so that's a cultural piece that I've carried with me from Merck, not something people might recognize from the inside, but some of my experiences working with you and others.
It's too bad we're on a podcast and we don't have video, because I have a huge smile on my face, Karen, just listening to the story. It's great, and it's been fantastic to watch you develop and contribute to our mission. It's been wonderful. And as I've gotten older, I've learned to balance some of my tendencies even, because it is great to come in and want to try to drive change and try to be dynamic. What I've learned is that organizations will naturally try to block that. If that's not the kind of foundation of those organizations, and most biopharmaceutical companies are not like that. Most biopharmaceutical companies by definition are not progressive. We're not like the tech industry. Right? Our life cycle is not months, it's years.
Yes.
It's very difficult to change the direction, but you can. You have to be consistent, right? You have to be repetitive at some level. You have to, we were just talking earlier, you have to be optimistic.
Yes.
Yeah, or you can't get stressed, you can't get beaten down. But you also need to deliver.
Yes.
And I think what I've learned in my infectious enthusiasm and exuberance to make things happen and do things differently, what I've learned through failures is that it's not just about doing something differently. And of course, this is obvious, but it's about doing it differently and being successful.
Indeed.
That's what you have to figure out, is how you thread that needle. And coming back to the talent piece, that's been the balance that I've learned. In the current job, one of the things I feel perhaps most proud about, and maybe in the same way that you might, given what you just described, is bringing people on who are a little bit more junior, who have all of the potential. You have to be really careful when you do that.
Yeah.
You have to have a lot of conviction. You have to be willing to invest
Right.
in their development. But those are the people that will change the world.
Right.
But then you need to surround them with some people who are experts in their domain.
Exactly.
Right, that aren't going to necessarily change the world, but that will ensure that we're able to execute and deliver.
And take informed risk, right?
Yeah.
I think that's where that juxtaposition is helpful, is when you've seen enough failure, you know that there are things you can see and there are things you can't see. And sometimes, I guess, when you're early career, you don't have that experience. But bringing those two worlds together, I think, is often quite powerful.
Absolutely.
Yeah.
I was in a leadership development course or something 20 years ago, and we did pre-work. I don't remember what the tool was that we used. We did pre-work where we filled out questionnaire, and it talked about our proclivity for change. And we created this continuum of individuals, or 30 or 40 of us, and we lined up. And on one end you had high score change, and the other end you had very low score change. Please never change anything. Please change everything all the time.
Right.
And at the end, the facilitator said, what you want to do if you're on the left side of the room is you want to become really good friends with people on the right side of the room.
Right.
So that when you're together, you work together to enact change.
Brilliant. So you've been doing this for how many years now?
Yes.
30-ish.
It's a long time.
It's a long time. You mentioned some of the things that you're proud of, and thinking back on programs and targets that you've worked on. Is there a particular favorite that you have where you were able to, with teams, transition from uncertainty to having a really outstanding result for patients?
Yeah. Well, so I'll give you the answer, but I'll tell you my first experience in 2001 when I came into the industry, a week after I had joined. So I told you I joined October 11th, 2001. This is October 18th, 2001. Right. And I'm entering an office. I'll throw out the names. I don't know if people will know them, but Barry Gertz is leading our clinical group. Paul Deutsch is my mentor and boss in clinical pharmacology. And Gary Herman is presenting his first study. It's a phase 2 study of a PDE4 inhibitor in asthma. And Paul brought me into the office, so it's the four of us, and I'm a fly on the wall. And Barry was a very intimidating figure, as you'll remember. And Gary presents the study, and it's like I've never seen a cleaner negative result than what he had in this study. It was so obviously negative. And I'm thinking, oh man, this doesn't end well. And he finishes presenting, and Barry gets up and stretches his hand across the table and shakes Gary's hand and says, "Congratulations, you just made a no-go decision."
Awesome.
And it was at that moment I realized in our business, of course, we need to see success. But success is not just a positive result. Success is an ability to make a clear decision and move on. So that's a mantra that's really stuck with me, is when you run an experiment, ensure that you have clear criteria and that you're able to make a decision at the end. We don't always end in that space, but that is our recipe for success.
Absolutely. Yeah.
But my favorite program is one that we're still working on right now at Takeda. It's an orexin 2 receptor agonist program for type 1 narcolepsy, but also for probably more than type 1 narcolepsy, probably for a range of sleep-wake cycle disorders. And I say this because the science is driven by genetics. Type 1 narcolepsy is a genetically predisposed disease.
Yeah.
The pathophysiology is so clear. The approach is very obvious. The way you get there is really difficult. And we've been working on this program since before I arrived at Takeda. So it's a program that's been ongoing for 15 years. I've been a part of the last 10 or 11. And when I came, it was somewhat of a twinkle in the eye of our discovery colleagues. And I got very interested in it because it scientifically was quite exciting. And the scientist in Japan who had started the program, it wasn't kind of on a big strategy slide. He was in his own lab working on it because he had a friend with type 1 narcolepsy.
I see.
And Merck had developed an orexin 2 receptor antagonist for sleep. And so now, of course, a centrally acting agonist is a bit more complex. And he had gotten one hit out of the screen. So there was no in silico approach at the time. It was all wet lab, and he got one hit, which is, I mean,
it's tough.
Absurd to start an optimization program on a hit, and he did. And we were able to bring it forward. And we're now actually on our 4th molecule. So not uncommonly, you have learnings and failures along the way. We had a very big safety issue with one of our oral molecules, and so now we're on our 4th molecule. We're in phase 3. That study should read out in a few months, and the phase 2 data look, transformative for these patients. So it's the combination of really extraordinary validation based on human genetics, incredibly rigorous and difficult path through discovery and development, and then, knock on wood, I think it's going to work, we'll see in a few months, the potential to fundamentally transform this disease for patients. And you don't get too many of those in your career.
Now, you said human genetics, but if I recall correctly...
Dog
genetics. Yes, isn't this the falling over animals who
exactly.
just fall asleep all of a sudden,
yes.
and people figured out that there was a gene involved, and those sorts of strong pathway biology stories are fascinating. And if you go back and you look, actually, I think there are those rare allele type of events that, as you said with HMG-CoA reductase, they actually show up later on now that we have these powerful tools to actually interrogate genetics more broadly.
How about you? Can I throw the same question back to you?
Yeah, sure.
What question, I mean, you still have many years left, but what if, as you look back in your career, what program has been the most exciting for you, or most meaningful, let's say?
Yeah, it's interesting that you point to the example where Gary presented. So, as you know, I came from the discovery world, and for me, the idea of following a molecule into the clinic and watching what could happen, it's about learning. And one of my favorite programs is not necessarily one where there was a powerful result, although Keytruda, as you recall, we were fortunate to be at Merck when the Keytruda moment happened, and you just saw the power of that mechanism unfold. And I remember a story of one of the patients who was almost at the last stage of his life, came on the trial and ultimately ended up being cured. And had a license plate made for his car that was Keytruda.
Oh, wow.
I mean, that's the power of someone who was writing wills and saying goodbye and being able to bring that person back to have a full and healthy life. Those are the amazing moments that inspire us to continue doing this despite the failures. But I will say that from a learning perspective, the trial that I was on, it was an asthma trial where we had seen beautiful PK/PD. Everything looked as if it was going great. And then in phase 2, the signal wasn't strong enough. Not only was the signal not strong enough, when you looked at standard of care that was already available, it wasn't going to have the kind of impact that we wanted. So we ended up terminating the program. Now, why is that important? Because it's easy to get very excited about the biology, the genetics, the PK/PD correlations, but at the end of the day, it's all about that efficacy. And the complexity of where our science then goes as it relates to patient population, marketplace, physicians, payers, and regulators. Even the most terrific science can get lost almost as it starts to transition through those real-world situations. And so that was an interesting learning for me, that even though we had an amazing package, it wasn't enough.
Yep, it's incredible. And as you're saying this, the world is becoming more complex.
Yes.
And the value of having that vertical input early into programs, especially understanding the commercial and reimbursement landscape that you're entering into.
Yeah.
In 2025, it's so much more critical than it's ever been in our history. But what I found, because I always follow the science, but of course you need to evolve the science around the other pieces. How developable is your program? And then of course, what does the commercial landscape look like? What we need to do is work together collaboratively, not to get a number, not to get an NPV,
right,
because they're meaningless, but to have that sophisticated input. So the NPV itself is, to me, it's problematic in the early space. It's fine as you move into late development, as you're thinking about business transactions, it's critical. In that early space, what's important are the assumptions that go into the NPV and understanding how those play out. That's how you can really drive decision making.
Absolutely. It's almost like your high change, low change, bringing those two people together.
Absolutely.
At least listening to each other, in this case, listening to the payer perspective, the patient perspective, the physician perspective, as you're sort of working up these programs, I think can give you amazing insight. And I encourage also that on my teams that even though you're thinking about the molecule, you've got to think about the patient
Absolutely.
and the outcomes at the end of the day.
Yeah. If you can not start a program versus carry one forward for 8 years only to stop it because you don't have a commercial path forward, I mean, think about that opportunity cost.
Yeah. So you've lived in Paris, you mentioned, San Francisco. Here we are in Boston, New York for a little bit.
Yep.
Do you have a favorite?
And I haven't lived in Tokyo, but I spent a lot of time in Tokyo.
Beautiful place.
Yeah,
Amazing.
yeah. So, it's such a good question. I haven't. It's almost like asking of your kids, do you have a favorite?
Yeah.
And maybe at any one moment you do. No, I think they all have their own special elements to them. I wouldn't say that I do. I mean, Paris it's a museum. It's the most beautiful, to me, the most beautiful city in the world. The hard part of living in Paris in our business is Paris, like a museum, doesn't change, right?
True.
And when I was at Sanofi, I used to bounce back and forth between Paris and Boston, and Boston is just so dynamic, and it's the epicenter of biotechnology, and so that's just so exciting. I don't have a favorite. Do you?
No, I'm a global citizen. I love, so it's very similar to you. As you know, I grew up in the UK, spent time on the West Coast in San Diego, New Jersey. We didn't mention New Jersey as a place.
Thankfully.
But,
strike that from the record, please. By the way, when I don't know how fully qualified I was for the job I'm in now when I was recruited into the job, because I didn't have as much experience in certain parts of the value chain that I do now, let's say. But I do know that one thing that was important for Christoph as he was thinking about the future of this company was to ensure that there was an R&D leader who did understand those cultural sensitivities of living and working in different environments, to your points earlier.
Yeah.
And he did say to me years later that had I not had the Sanofi experience, had I not lived as an expat abroad in a different culture, that he wouldn't have hired me for this job.
That's very interesting.
Yeah.
I mean, this is a global endeavor in a way, right? We work, even if a company is based in one city, biopharmaceutical research is really a global endeavor. There's collaborations either on the biotech industry side or academia. That's something I recall working with you at Merck. We had a lot of collaborations. Even if you're not necessarily from a sort of diverse background in terms of where you've lived and worked, I think by default, our industry sort of forces us to be
Absolutely.
In this sort of mode of working in a global sense. So we are at sort of an intersection here where the traditional biopharmaceutical research, lab-based research, which I think will always be part of how we do what we do, clinic-based research, is now intersecting with technology. How do you look at that sort of intersect, where we are today, where we're going? Do you have any predictions for how AI and machine learning are going to transform or not transform what we do?
I'm on the transform side of the spectrum.
Okay.
I don't think even the skeptics, I don't live in their minds actually, so I don't know their level of skepticism. I sense they react to some of the extreme places that certain people go. And I agree with you that I don't ever see a time, certainly not in our lifetimes, but probably beyond, where wet lab science and running clinical trials in human subjects is not going to be a fundamental part of what we do. But in both discovery and in development, there's so many inefficiencies that are primed for advanced technologies to help us with. I mean, just automation.
Yeah.
You know, if you look at generative AI all of the document work that we have to do and all of the repetitive activities that we have to do. If you just look at a clinical trial and you look at the dossier from the trial and the investigator brochure to the CSR and all of the documents that come out of those three,
Yeah.
I mean, that's your whole regulatory filing, essentially. And if we can do more to automate that process, it will help us with insights,
Yeah.
but it will certainly speed up significantly and reduce the amount of costs it takes to generate. So that's just operational. I think there are massive changes operationally. And then, of course, discovery is more challenging because biology doesn't always follow logic and laws in the same way that physics does. But I still think that and we've seen this play out, there's a huge opportunity to simplify discovery, reduce cycle times, and reduce costs. So I think I'm in the transformative camp, and to me it's just a question of how long, not if.
Yes, agreed. I think, as you point out, biology is sufficiently complex that we're still very much in learning mode. I think that the ability to automate, to run more experiments, to learn from what the data, the complex data, is telling us, I think there is going to be some transformation there. But even after that, we still have to be able to decide, is this the right target for the right patient, for the right indication? And that requires the kind of integrative thinking that humans are really good at.
Exactly.
So I see it more as an assist than anything else.
Excellent. I like that. I like that term.
Assisted intelligence, not artificial.
Exactly.
So what about you as a person? Are there things that we don't know about you? Music, sports, things that motivate you beyond trying to cure disease?
Oh, for sure. I'm curious. Curiosity is one of the characteristics I've carried with me through my whole life. And in fact, I've learned, either because it's self-serving or because it's real, that it's a really healthy leadership trait. If you're really curious and you're really interested in what people are saying, you learn so much about your organization that you're leading, and it helps you so much lead in a way that's not just about your vision, but about what your organization needs. It's also engaging. People like it when you're curious and engaging with them. And so this trait started when I was a young boy, and there are two moments that my parents, my mom who's still alive, my dad who died a couple of years ago, would remind me of often. The first was my mom as a 3-year-old. I was one of 5, but I was 2 older brothers, an older brother and an older sister who was 6 and 7 years older than me. So I was almost like a kind of a youngest child. And then my parents had twins, mistakenly had twins 3 years later. Okay. They're not mistakes in the end, but it was not planned. And so at age 3, I would follow my mom around in the garden and just ask question after question after question. And one day she stopped, she paused, and she looked at me and she said, "Andrew, it's time for you to go to preschool." And then my dad, asking question after question. And he was a bright guy. He used to love answering my questions. And I said once, "Dad, if I had someone who could answer every question in the world and that person was available to me for the weekend, I wouldn't be able to ask as many questions as I have." Really? So curiosity is a really big part. And so that leads me to roam in life, sports, music, literature, history. I love everything. And I find that that's something that keeps me going. So I had a boss, Elias Serhouni, at Sanofi, and he gave a convocation address at MIT a number of years ago. And his theme, I think, was a brilliant theme. It was a 50-50 theme. 50% of life do X, 50% of life do Y. And he talks about reading and learning. And he said, to make yourself the best that you can be in whatever job you're doing, 50% of your reading should be about that technical subject matter, and 50% should be about everything else. And you're able to then integrate different worlds with one another.
Brilliant.
Yeah.
And in this day and age where I feel like the next generation perhaps doesn't read as much, I worry about that because I love reading. In fact, I'm scolded often to go to bed because I want to read. I have to do it every single night.
What do you read?
All sorts of things, to your point. I mean, I also love history. Unfortunately, I don't have as much time to read about history as I would like. I'm often reading papers about targets and diseases, but I feel as if the next generation perhaps isn't as immersed in that serendipitous learning that you get from reading. Do you agree with that?
100%.
Yeah.
100%.
I don't know where that goes.
Yeah, I find, we were talking about stress management as another leadership trait that's so important. And I find that, of course we all get stressed at some level, and it's managing through that stress because our jobs are really tough and the stakes are really high.
Yeah.
And I find that I'm best with myself and my stress when I'm reading.
Yeah.
And I don't know if it's causal or not. But when I'm not, I find that when I'm not reading, and oftentimes I realize that in retrospect, when I'm not reading outside of medicine and science, I find that I find life harder. It's a bit more stressful. Yeah.
That resonates.
Yeah.
I often share poetry with the team.
Oh, is that right?
Yeah, which my kids think is really weird. But finding quotes or excerpts from poems and then finding the full piece and understanding why that quote and how it fit into the whole poem. I love doing that.
It's brilliant.
My dad, he used to say it's better to be envied than pitied, for example, and I ended up finding out where that came from. Both my parents actually are really good writers. In fact, my mum writes poetry, so for me it comes very naturally to reach out to that as a resource.
That's great.
Yeah.
One question that you sent to me in advance of this podcast that you asked me to think about is there one book or something that I've read or song or that's really been particularly meaningful to me, and I've thought a lot about it and I struggled with it because there's so many.
Yes.
Right, pieces, but there is one actually. It was an op-ed piece that was written by David Brooks maybe 12 or 13 years ago that I was combing through the New York Times and I landed on it, and it was about, I can't remember the name of it. Maybe it was about your eulogy CV. So there was your professional resume, which is of course everything that you've done, where you've gone to school, the jobs you've had, the accomplishments professionally. Yeah. And then there was your eulogy CV, which is what people really remember. It's not so much what you've done, but how you've made them feel and how you've treated them.
Beautiful.
And the person that you are.
Yes.
And I remember that so well because I read it and reread it. In the world that we live in today, there's so much happening because it talks so much about us as people, and how important it is for us to be human and to treat people as humans. And I went back to it recently because we're in the world that we live in where there's just so much polarization that's not really reflecting who we are as people, and the differences that exist across political parties across the world. They're not as great as they're made up to be. In the end, as people, we're much more similar than dissimilar. And I was reminded of that in this David Brooks piece. I would really encourage you to go back and read it. It's incredibly inspiring.
I am going to find that.
I'll send it to you.
Yes, please.
Yeah, okay.
Yeah, and I think that's one of the things about travel and working in different places. We have an office in Hyderabad in India, and when you go to these different places, you are reminded that we are much more similar than we are different, right? People tend to think about something they don't know or understand or haven't experienced as different and problematic, but actually there's so much beauty in the similarities and also the small differences that we have, right? Andy, it's been so terrific to speak to you. I'm still inspired by those days 20 years ago, even as I work today. Your energy, your infectious enthusiasm, your curiosity, those have all stayed with me. And so it's just been wonderful to sit down with you and talk through what we do for a living, some of the struggles we face, but also the hope and optimism that we can still help patients once in a while.
Thank you very much, Karen.
Thank you.